Hello. Today we'll continue our lecture about these topic techniques and applications of recombinant DNA technology. This slide summarizes all of the technique. Usually recombinant technology is used to express protein and study their structure and function, as well as study gene structure, sequence, and expression. They can be used. Biotechnology can be used to create transgenic animals and gene knockout mice. As well, it can scale up production, isolation, and purification of therapeutic proteins, DNA products. As well, it can be used in human gene therapy. The purified protein we can make from it antibodies for medical purposes. We can as well find the chromosomal location of cloned genes and detect abnormalities and copy numbers. As well, we can use it to mutate a gene and study function in the altered protein produced. And create new genetically microorganisms and animals can be used in several applications. As well, we can use biotechnology in forensic applications such as DNA printing, and we can use it for diagnosis of human genetic disorder and infectious disease conditions. First technique is Agarose Gel Electrophoresis. I'll go fast on that because most of you are familiar with that. It's actually separate and visualize DNA fragments based on size. Is isolated. This agarose is a sugar isolated from seaweed, and when melted in buffer solution and left to cool, it will form a semi-solid gel containing small pores like a mesh through which DNA will travel. The percentage of agarose used to make the gel it depends on what you need to separate. Usually the percentage from 0.5 to 2% max. The high percentage allow resolve smaller size of DNA fragments, while the low percentage resolve larger size. To run a gel, it is submerged in buffer solution that conducts electricity. DNA is loaded into small depressions called wells at the top of the gel. The electric current is applied through electrode at the opposite ends of the gel, and then DNA migrates according to its charge and size. The rate of migration through the gel depends actually on the size of the DNA. The migration distance is usually inversely proportional to the size of the DNA fragment. DNA migrates toward the positive pole. That's because the total charge of the DNA is negative, so that's why it migrates towards the positive pole due to the fact that the sugar phosphate backbones make it always negative charge, so larger fragments migrate slowly and vice versa. We usually use two types of dyes, a tracking dyes and the DNA staining dyes. The tracking dyes is adding to the sample to monitor migration visually by eye, while the DNA staining dye, that we need this dye to visualize the DNA after the electrophoresis. One of the DNA staining dye is ethylene bromide. It intercalates between DNA base pair and it fluoresces under UV light. Then we can take a picture for this DNA. But keep in mind that ethylene bromide is highly carcinogenic, so the scientists now are turning to use other compounds that are less harmful. This is a represented figure showing the agarose gel electrophoresis. You can see mixture of DNA fragments of different size. They are loaded into the wells. The wells is located onto the cathode, and then once you switch on the power, the DNA fragment starts to migrate. Each DNA fragment with the size is represented by a band. You can see the band, the blue bands. The longer fragments are retarded in migration, while the shorter fragment are faster in migration. And then you can visualize band by fluorescence under UV light. You can see this is actual image of a DNA agarose gel of DNA have been cut by different restriction enzymes. And you can see we usually use a marker that is known base pair size, so to compare the size with the size of the bands with the marker. So restriction mapping can be used for sequencing. So we can digest a clone gene, the DNA fragment, with a single or double restriction enzyme. Then we separate DNA fragment via agarose gel. So you can see from the figure the difference by using the restriction enzyme, and when we use one single restriction enzyme, and then when you use double restriction enzymes. If you can see, this is the DNA which have been run on the agarose gel. So you can see there is two sites for the restriction enzyme. So once you cut by BAM alone, it will generate two sites two fragments of DNA. Once you cut by PST1 alone, it will generate two sizes as well, two sizes of DNA as well. But when you cut by both DNA fragment, it will generate three different sizes represented on the agarose gel by three bands. The second technique is chain termination DNA sequencing. Normally we call it Sanger method. This is important, of course, to determine the sequence of nucleotide of the cloned gene. These two, we use it to know the nucleotide sequence of cloned gene of expression, to determine the exact structure of a gene, to identify regulatory elements such as promoter, to identify differences in gene created by gene splicing, and to identify genetic mutation. What about the reaction? The reaction consists of several components. First component is the primers annealed to the denatured DNA template, the four normal deoxyribonucleotides, and the DNA polymerase, and the special component which is called DDNTP. This DDNTP, actually, the 3' OH is replaced by 3' H, making this group is non-reactive. So once this DDNTP is being added to the polymerization is terminated. Over time, this DNTP will be incorporated in the position, and as I told you, the chain will be terminated. Then the fragments are separated by polyacrylamide gels, and then you do autoradiography because this nucleotide is radioactive labeled to check them, and then you read the gel from bottom to top. The sequence generated from the reaction is complementary to the sequence on the template strand in the vector, because actually the polymerization is complementary to the template. There is a quick assignment you need to do in one page in a Word file, one page in a Word file, and just from over the internet, make this page about your opinion about the human genes or gene sequence can be patent or not. There is a controversy in this issue. For a figure, you can see this figure summarize the Sanger method. So you can see actually in a test tube, you have the first component is the template, and then the primer, and then the DNA polymerase, and then you have the normal DNTP, the four different type of DNTP, and then you have the special one is the DDNTP. You can see that it looks different. So once there is a polymerization in the bottom figure, you can see once at any time, once this DDNTP is added based complementary on the template, the chain is terminized. There is a termination of the chain, and all this fragment which is formed is labeled. You run the gel on polyacrylamide gel, and you can see these chains, this fragment which is formed of different lengths will migrate through the gel according to size. If we look closely from bottom, the sequence, you can know the sequence of the nucleotide if you enlarge this. You can read from the bottom based on the label on the above. So the first sequence is C, then G, then A, then TT, then G, then A, okay, and so on. But keep in mind that this sequence actually is complementary to the template strand, okay? And as you can see, it starts from the 5'. Okay, so you have a laser. Another way of detecting, instead of doing the gel, you if the nucleotides have been labeled with a fluorescent dye, you have a laser and detector, and that converts the signal into computer signal, and you can see the sequence arise as a peaks. That's a computer automated DNA sequencing. The next technique is NGS, or next generation sequencing. It's the advanced way of sequencing DNA. These produce highly accurate and long stretches of DNA sequence, more than 1 gigabase of DNA per reaction, low cost, up to 200 times faster than Sanger. Roche, it's a company. Roche Roche as a company produced Roche 454 Life Systems is the first company to commercialize the NGS technology. The process actually is called solid phase method. You get the fragment genomic DNA are attached to beads loaded into multi-well plates mixed with the DNA polymerase fragment or PCR amplified in a separate water droplet in oil for each bead. It's like a multi-well plates contain more than 1,000,000 well with one bead per well. Each serve as a reaction tube. So instead of doing in a million BCR tubes, small BCR tubes, you do it in a million well, in a multi-well plate per reaction. This reaction is called biosequencing. The reaction biosequencing is then used to sequence DNA on the bead in each well. In this reaction, a single labeled nucleotide, DGTP, is flowed over the well. So a single labeled nucleotide one at a time over the well. Each well contains a single bead along with primers annealing to the DNA on the bead. When a complementary nucleotide crosses a template strand adjacent to the primer, it is added to the 3' end of the primer by DNA polymerase. Incorporation of the nucleotide results in the release of pyrophosphate. This pyrophosphate, which is the PPI, initiates a series of light-releasing reaction, chemiluminescence reaction, that produce light by using enzyme luciferase. So actually, each nucleotide will generate a different light, okay? A different light based on the tag of the fluorescence it have, okay? This emitted light from the reaction is captured and recorded to determine when a single nucleotide has been incorporated into a strand. By rapidly repeating this nucleotide flow, so they flow the nucleotide one time at a time. If this nucleotide is complementary, it will bind, it will emit a signal. This signal, its color, will tell us from the detector which type of nucleotide. So you can see this figure summarize the principle. You can see that in each well there is a bead, and this bead, the DNA template attached to it, DNA on bead subjected to the biosequencing reaction. You can see on the bead they have the template, the primer anneals. You add the nucleotide with the label. It generates label. Like look down in the figure at the bottom. You can see the G generates PPI, and this PPI in the presence of luciferase enzyme will generate light, and it can be detected. And this is the NGS technology. The next technique is fish fluorescence in situ hybridization. In this technique, we can detect used to identify a gene of interest and in which chromosome. Chromosomes are isolated from cells, spread out on glass microscope. Use DNA or RNA probe for the gene of interest, complementary to the gene you are looking for. These probes are fluorescent tagged and incubated with a slide. This probe actually will hybridize with the complementary sequence on the chromosomes. You wash the slide and expose to UV light. Wherever probe has bound to the DNA, to the chromosome, it will eliminate the signal for its presence. Then you do karyotyping to spread the chromosomes over a slide, and you can see from the bottom figure that you see chromosomes with spots. That's mean it's a signal. So what does this fluorescence on more than one chromosome indicate? Indicate the existence of multiple copies of gene and the related sequence that may be a part of the gene. So what is the application of fish? Fish can be used to analyze genetic disorders, can be used to determine which cells in particular organs are expressing the particular messenger RNA. The next technique is Southern blotting, and this used to determine gene copy number, gene mapping, gene mutation detection, PCR product conformation, and DNA fingerprinting. Actually, the procedures, you digest the DNA into small fragments with restriction enzyme, separate by agarose, treat with alkaline solution to denature DNA, melting DNA. Fragments are transformed into nylon or nitrocellulose filter membrane. This is called blotting. This filter or the blot is exposed to the UV to permanently attach the DNA. The blot is incubated with the probe. It is a sequence of nucleotide that is complementary for your gene of interest. and they expose the film by autoradiography. The number of bands on film represents the gene copy number. How we can study gene expression? Techniques involving analyzing messenger RNA produced by tissue. You can use either Northern blot analysis, reverse transcription, RT-PCR, qPCR, in situ hybridization, or gene microarray. So if we continue the southern blotting, you can see here that you have a gene of interest that have an EcoR1 site in the middle and the BAM-H1 at the end. So once you do restriction enzyme, and the first one is labeled DNA marker of known size, and the second one you do restriction by BAM-H1, and the third one you do restriction with EcoR1. So actually EcoR1 is cutting inside the gene, while BAM-H1 doesn't cut inside the gene. Then you run agarose, and you transfer on a nitrocellulose membrane or nylon the blot. Then you take the blot and expose to radiography, and you can see actually the second lane shows that it is being cut by BAM-H1, but since BAM-H1 have no site inside the gene, so still the gene of interest is one single piece. But when we cut it in the third lane, when we cut it with the EcoR1, you can see the DNA is digested into two fragments. To study gene expression techniques involving analyzing messenger RNA expression, you can see here from the plot, you can see from plot 1, these are actually 3 plots of an autoradiogram from a northern blotting experiment. In the first one, you can see in which RNA from 4 rat tissues, that's a 4 rat tissues, this RNA from the seminal vesicle, 2 from the kidney, 3 and 4 from the epididymis. Okay. So you can see when you run agarose gel from RT-PCR experiment, in which RNA from rat tissue was reverse transcribed and amplified with primer of actin for actin defensing. Northern blotting. The basic method is similar to southern blotting, and they can compare the quantity amount of messenger RNA present in different samples. The procedure actually, you isolate the RNA from your tissue, separate by gel electrophoresis, blot on the membrane, same membrane, hybridize by probe, same principle, expose band on autoradiography, and show presence of messenger RNA for gene of interest as well. So actually, northern blot is for mainly for RNA, while the southern blot for DNA. RT-PCR actually is used to study messenger RNA level when level of detection is below that of northern blotting. The procedure actually pretty the same, except you're doing PCR, isolate messenger RNA, do reverse transcription to make double-stranded cDNA, use PCR with a specific primer to amplify the gene of interest, run Agarose gel to separate amplified fragments, and then you determine the expression pattern in the tissue. The amount of cDNA produced on RT-PCR reaction of a gene of interest reflects the amount of messenger RNA and the level of gene expression. Thank you. Real-time PCR or quantitative qPCR. These techniques can be used to quantify amplification reaction and they occur in real time. They need a special thermal cycler that uses a laser to scan a beam of light through the top or the bottom of each PCR reaction, and each reaction contains either a dye containing a probe or a DNA binding dye that emits a fluorescence light when illuminated by laser. The light emitted by the dyes correlate with the amount of PCR product amplified. Then the light is captured by a detector and the amount of fluorescence is read by a software. The readout is plotted and analyzed to quantify the number of PCR product produced per each cycle. There are 2 approaches. Either use a Tachman probe, and in a Tachman probe, it's actually a short sequence of DNA which is complementary to a specific region of target DNA between forward and reverse primer. This Tachman probe contains 2 dyes, a reporter dye at the 5' end and the quencher dye at the 3' end. And the other one is cyber greens, which binds to, it's another type of dye, can bind to newly formed double-stranded DNA and add more, as more double-stranded DNA is copied with each round of qPCR, there are more DNA copies, so they are more cyber green, which emit lights under fluorescence. which emit fluorescence under the UV. This diagram summarizes the steps of a qPCR. You can see that the primers, forward and reverse primer, anneal. Then you have another primer which have the reporter and the quencher at the 5 end and 3 end respectively. Soon after that, the polymerization starts from the forward primer and the reverse primer. Once the polymerization reaches a reporter molecule on the 5' of the probe, it will start, upon the excitation of UV, it will start to make fluorescence. And here you can see how it is read out on the computer software, so you can see how the curve looks like for high gene expression and low gene expression. In situ hybridization is a type of hybridization that uses a labeled complementary DNA, RNA, or modified nucleic acid strand, like probe, to localize a specific DNA or RNA sequence in a portion or section of tissue in situ. Gene microarray. Gene microarrays enabled researchers to study all the genes expressed in a tissue very fast. This gene chip, the microarray, is created with use of small glass microscopic slide. The single-stranded DNA molecule are spotted on a slide using an arrayer, which is a computer-controlled robotic arm, which fixes DNA or multiple copies of cDNAs at different spots on the slide, and which is recorded by a computer. The procedure actually extracts RNA from a tissue of interest and does a cDNA by reverse transcription from the messenger RNA, and you label with fluorescence dye. This labeled cDNA is incubated overnight on the array, where it hybridizes with different spots on the array that contain complementary sequence. This array can have more than 10,000 spots of DNA. The array is washed and scanned by laser that causes cDNA to hybridize to the array. The cDNA which already hybridized to the DNA which exists on the array will fluoresce under excitation. These fluorescent spots reveal which genes are regulated, and the intensity of fluorescence in this indicates a relative amount of gene expression. These figures summarize what's happening in the microarray. So we first extract RNA from the tissue sample, then you do reverse transcription number 2 to get the cDNA, then you incubate this cDNA molecule, you do it a single strand, melt this cDNA molecule, and then you get the array, which looks like the figure on the top right. You can see incubate this single-stranded cDNA with the array, which have another a lot of 10,000 spots of cDNAs. So once this cDNA have a complementary sequence on the array, it will bind, and once you excite light on it, the computer software will detect based on the amount of light if it's a bright fluorescence, highly expressed gene is present. If it's no fluorescence, there is no expression of that gene. If it's moderate, this means low gene expression. Gene mutagenesis studies. It's used to study the structure and function of protein produced by a specific gene. Site-directed mutagenesis is used to create specific nucleotides of a cloned gene contained in a vector. Genes is then expressed in cells, which result in translation of mutated proteins. So why use this approach? The procedure lets researchers study effects of mutation on protein structure, as well as provide information about which nucleotides are important for specific function of a protein. RNAi. Naturally, these RNAis are naturally occurring mechanism for inhibiting gene expression. Its procedures, actually, the double-stranded RNA can be bound by a dicer enzyme that cuts the double-stranded RNA into around 25 nucleotide fragments, and these are called small interfering RNA, siRNA. This siRNA can be bound to a RISC RNA-inducing silencing complex. This RISC unwinds, melt the double-stranded RNA, releasing single-stranded RNA that can bind to messenger RNA of your gene of interest. The binding of siRNA to this messenger RNA leads to the degradation of the messenger RNA by a silencer enzyme, or blocks the translation by interfering with ribosome binding. Currently, there are many developing techniques to use RNA to silence gene expression. This figure summarizes how RNA works. So you have a double-stranded RNA with the aid of the dicer, and the dicer will break this long double-stranded RNA into short ones. Then the RISC, including the sizer, will melt this RNA into small fragments called siRNAs. This siRNA will bind complementary to the messenger RNA of interest. You want to knock down it or down-regulate it. So it will either degrade messenger RNA by silencer or inhibition of messenger RNA by interfering of the binding of the ribosome to the messenger RNA. The second topic is genomics and bioinformatics. Actually, it's a hot discipline in biotechnology. Genomics actually it's a cloning, sequencing, and analyzing entire genome by a process called shotgun sequencing or shotgun cloning. They use restriction enzyme to digest pieces of entire chromosomes. The procedure will lead to thousands of overlapping fragments called contigs, which are each sequence. So you have a lot of contigs, you sequence each of them. The computer program will align these sequences based on overlapping sequences pieces. Bioinformatics, actually it merged the molecular biology with computer technology. This interdisciplinary field applies computer science and information technology to promote an understanding of biological process. Its application actually used it as a database to store, share, and obtain the maximum amount of information related to gene structure, gene sequence, and expression, and the protein structure and function. The gene bank actually it's a public database of DNA sequence and contains all the National Institute of Health collection of DNA sequences. Each entry has an accession number. Each entry of the genes of the sequence of the gene has an accession number that scientists use to refer back to the clone sequence. These are maintained by the National Center for Biotechnology Information, which is NCBI. Basically, actually, it's a PubMed research database that you use to search for any paper you want to read about research. NCBI is a goldmine for bioinformatics resources that creates a public access database and deploys computing tools for analyzing and sharing genomic data. You can see here how we do this sequencing. You can see that the genomic DNA can be cut by a number of enzymes. A lot of fragments will arise. This collection, we call it library or contigs. These contigs can be aligned used by computer database to determine the sequence of these pieces, and they align to know the unknown sequences. And here you can see how they overlap the sequence to see the complete sequence of a DNA of a specific region. They can use as well a DNA database searching tool, which is BLAST, Basic Local Alignment Search Tool. This BLAST actually they compare the sequence with other sequences in other organisms to see if it's a complete match or no match. So in this figure you can see the human OB gene is very similar to the mouse OB gene. The Human Genome Project. Starting in 1990 by the US Department of Energy, in April 14, 2003, the human genome have been completed, MAP. The international cooperation of efforts to identify all human genes and to sequence all the base pairs of human chromosomes, mostly done by 20 centers in 18 countries, as you can see. The Human Genome Project HGB designed to accomplish the following: analyze genetic variation among humans, that means SNPs identification, map and sequence the genome of model organisms, including bacteria, yeast, roundworms, fruit flies, mice, and others, develop new laboratory technology such as high-power automated sequencers and computing technology, as well as widely available database for genome information, which can be used to advance our analysis and understanding of gene structure and function. It is used as well to disseminate genome information among scientists and general public. They consider the ethical and legal and social issues that accompany the HGB and genetic research. So what we have learned from HGP, nearly 15% of genes do not yet have a function. The summary of the finding of the HGP project, 3.1 million base pair make up the human genome. The genome have approximately 99.9% are similar between individuals of all nationalities. There is a lot of SNPs or CNV copy variation number, such as the long deletion, insertion, duplication in the genome, and account for much of the genome diversity identified between humans. Less than 2% of the genome codes for the gene. The vast majority of our DNA is non-protein codon, and there is a lot of repetitive DNA sequence account for at least 50% of the non-coding DNA. The genome contains approximately 20,000 protein coding genes. Many human genes are capable of making more than one protein. That's because, of course, of the alternative splicing, allowing human cell to make at least 100,000 protein from only about 20,000 gene. Chromosome 1, which is the biggest chromosome, have the highest number of gene, and the chromosome Y, which is the smallest chromosome, have fewest gene. Many of the genes in a human genome show a high degree of sequence similarity to genes in other organisms. Thousands of human genes have been identified and mapped to their chromosomal location. The top figure is for knowledge. You can see on the x-axis the number of chromosomes, and you can see on the left y-axis the number of genes it contains. On the right y-axis you can see the number of base pairs it contains. The Human Gene Project have started another expression called omics. It makes a revolutionary expression, omics, in which in proteomics it is studying all protein in a cell. Metabolomics is studying proteins and enzymatic pathway involved in cell metabolism. Glycomics is studying carbohydrates of a cell. Transcriptomics is studying all genes transcribed in a cell. Metagenomics analysis of a genome of an organism in an environment. Pharmacogenomics customized medicine based on person's genetic profile for a particular condition. Nutrigenomics, interaction between genes and diet. Comparative genomics, it is the sequencing genome for a number of model organisms. Stone Age genomics, Genomics, is analyzing ancient DNA. This figure for knowledge is showing the proposed function of a number of human genes assigned to a functional category. And you can see from this figure, the blue area, that you have around 41.7% of the genes of unknown molecular function. This table as well is for knowledge, but it shows the comparison of some of the selected genomes. So you can have the organisms, the approximate size of genome, the number of genes that these organisms have, the approximate percentage of genes shared with human, and how we get this information through the internet. Thank you.